So a synapse is a site of communication between two neurons or between a neuron and an effector cell. So you have there your synaptic end bulbs. These are bulb-shaped structures that swell at the tip of some axon terminals. String of swollen bumps are called your varicosities. That they may contain some tiny membrane enclosed sacs called your synaptic vesicles that encloses and stores a chemical called your neurotransmitter. So a neurotransmitter is a molecule released from the synaptic vesicle that excites or inhibits another neuron, muscle fiber, or gland cell. You have different nerve fiber types. So type A generally are large myelinated, and you have subtypes. So alpha, these are motor, skeletal muscle function. Beta, you have sensory touch, pressure, vibration. You have gamma for muscle spindle, and delta for sharp, localized pain, temperature, and touch. Your B fibers, these are small, myelinated, functions more on the preganglionic autonomic. Then C fibers, these are for diffuse, deep pain, temperature, and postganglionic autonomic function. The conduction velocity is directly proportional to the cross-sectional area of the axon, so thicker fibers conduct more rapidly than those with smaller diameter. Your non-myelinated axons, or fibers, the action potential passes continuously and progressively exciting neighboring areas of the membrane, while your myelinated fibers, they have a positive or presence of myelin sheath as insulator, so only few ions can flow or pass through. So stimulation occurs only at the nodes of Ranvier, where the action potential jumps from one node to the next. So the current produces depolarization at the next node. This leaping or jumping action is also known as your saltatory conduction. Let's talk about your axonal transport. Your anterograde transport, so the action potential flows from the cell body towards the axon, while your retrograde transport, the cell body responds to changes at the distal end of the axon. Okay, so if there are changes or activities within the axon, your cell bodies will also have certain activities, but only within the cell body. The synapse is basically the space or the functional junction between your presynaptic and your postsynaptic neuron. So your presynaptic neuron, this is the nerve cell that carries and sends a signal or nerve impulse towards the synapse. While your postsynaptic cell or neuron, they receive a signal or carries a nerve impulse away from the synapse to an effector cell. Now let's differentiate between your neurotransmitters and your neuromodulators. First, your neurotransmitter. So upon the inflow of your calcium and attachment to the synaptic vesicles, it will release your neurotransmitter along the synaptic cleft. So your neurotransmitter will also bind with the receptor protein, that will trigger the opening of your ion channel. So basically, it utilizes your ionotropic receptor. This can either cause excitatory postsynaptic potential or EPSP or inhibitory postsynaptic potential IPSP. For the excitation, you have there your acetylcholine nicotinic receptors and your L-glutamate. While for inhibition, you have your GABA. For your neuromodulators, so the receptor proteins will bind with the neurotransmitter, but with the help of a second messenger system, your G protein. So basically, it utilizes your metabotropic receptor. So it has a longer latent period because it will depend on the summation of the graded potential that can either cause depolarization or hyperpolarization. So for example, you have your acetylcholine muscarinic receptors, you have your serotonin, your histamine, neuropeptides, and adenosine. So generally, they will enhance, prolong, inhibit, or limit the principal neurotransmitter's effect on the postsynaptic membrane. So for normal synaptic function to occur, you also need to remove the neurotransmitter from the synaptic cleft. So first, through diffusion, so if the neurotransmitter no longer exert an effect, so they will just diffuse away. Then you have your enzymatic degradation. So for example, your enzyme acetylcholinesterase would break down your acetylcholine. Then you have the uptake by cells. So the neurotransmitter transporters will transport it to the neuron that released them through reuptaking it, or into the neighboring neuroglia, by uptaking it. This usually follows a one-direction communication. So it depends on the site of the synapse. First, it can be axodendritic, meaning axon to dendrite. It is the most common type. Then you have your axosomatic, axon to cell body, or axoaxonic, axon to axon. You have two types of synapse. You have your electrical, which is usually direct, and chemical, usually indirect. First, your chemical synapse, with the presence of your gut junctions containing your channels or connexons, in which it extends from the cytoplasm of your presynaptic neuron to the postsynaptic neuron. It is usually bidirectional and usually common in your smooth muscles and cardiac muscles. So they have two main advantages. You have a faster communication through the presence of your gut junctions, and synchronization, since it produces your action potential in unison that they are connected by the said gut junctions. Then you have your chemical synapse. So the presynaptic neuron converts the electrical to chemical signal by releasing a neurotransmitter. Like for example, with the arrival of your action potential, this will open the voltage-gated calcium channels, causing your calcium ions to flow inside, binding to the vesicles, releasing your neurotransmitter. Then your neurotransmitter will bind to the receptors of your postsynaptic neuron, creating a postsynaptic potential. So it is usually unidirectional and with a slight delay of 0.5 millisecond. Spatial and temporal summation of neurotransmitters. So the integration of this input involves summation of the postsynaptic potentials. When you see the word postsynaptic potential, it means the action potential within the postsynaptic neuron. First, you have your spatial summation. So this refers to the summation of the postsynaptic potentials in which the stimuli occurs at different locations of a postsynaptic cell at the same time. Like for example, the buildup of neurotransmitter released simultaneously by several presynaptic end bulbs. Then you have your temporal summation. So this pertains to the summation of postsynaptic potentials in which the stimuli occur at the same location of the postsynaptic cell but at different times. Like for example, the buildup of neurotransmitter released by a single presynaptic end bulb firing rapidly 2 or more times. The sum of all the excitatory and inhibitory effects at any given time determines the effect on the postsynaptic neuron, which may respond in the following ways. First is your APSP, your excitatory postsynaptic potential. So this is where the excitatory effects is greater than the inhibitory effects, but it remains sub-threshold, meaning it is only partially depolarized. But with summation, that's the time it can permit or it can transmit your nerve impulse. Then you have your action potential or nerve impulse. So this is when the threshold is reached with excitatory effects more than inhibitory effects. So the action potential will be triggered. Lastly, you have your IPSP, your inhibitory postsynaptic potential, where your inhibitory effects are greater than the excitatory effect. So this will lead to your hyperpolarization, therefore it cannot transmit your nerve impulse. When your ion channels will open, this will allow specific ions to move across the plasma membrane down their electrochemical gradient. Okay, so with the chemical part of the gradient, so the ions move usually from areas of higher to lower concentration. Then the electrical aspect, usually positively charged cations will move to a negatively charged area, and negatively charged anions will move to a positively charged area. So the movement of ions will create a flow of electrical current that can change the membrane's potential or polarity. So you have different types of ion channels. You have your leak channels, which randomly opens and close, ligand-gated channels. You need a chemical in a form of your neurotransmitter, hormone, or ions that will open the gates. You have your mechanically gated channels. You need a mechanical stimuli. It can be vibration, pressure, touch, or stretching. Then you have your voltage-gated channels, in which this will open if there are changes in the membrane potential. So the difference between your resting membrane from your graded potential is that your graded potential occurs when there is a small deviation from the membrane potential. So it mainly occurs within the dendrites and the cell body. So as you can see, it only occurs in a short distance communication. With the opening and closing of your ion channels, this will alter the flow of ions across the membrane, leading to a localized current flow or a local current. So this occurs only in a short distance, then gradually dies. This is what you call your decremental conduction. While your resting membrane potential of a nerve is around negative 70 millivolts. So this is the resting state or the unstimulated state. So there is basically no movement of the ions yet. But since your potassium is readily permeable to the membrane, So there is a passive efflux or outflow of your potassium ions. This can cause change to the membrane potential. Then lastly, let's talk about the membrane excitation and conduction. So take note that the ions are abundant either inside or outside of the cell. So just remember that your sodium is more abundant outside and your potassium is more abundant inside. So let's recall your electrochemical gradient. So the chemical aspect of that gradient is that ions flow from higher to lower concentration. Since your sodium is more abundant outside, it tends to flow inside. So there is sodium inflow. Then your potassium is more abundant inside. That is why it usually flows outside. Okay, then you have your electrical aspect of the gradient. Since your potassium channels are naturally leaky, your potassium ions tend to flow outside passively. So this will cause a change in the membrane potential. So remember that positively charged ions will go to a negatively charged area and negatively charged ions will go to a positively charged area. Since with the exit of your potassium ions, in which your potassium ion is a positively charged ion, this will make the inside of the cell more negative. That is why this will increase the membrane's permeability to sodium. Okay, so this will trigger the start of your depolarization. This will occur as long as it can reach the threshold. The threshold is around negative 55 millivolts. Once the threshold is reached, then it can transmit or trigger the action potential. This basically describes the all or none principle. It's either the action potential occurs completely or does not occur at all. So the process basically begins when the nerve cell is excited. This can be stimulated by any electrical, mechanical, or chemical means or chemical stimulus. So with the potassium ion exiting the nerve, this will make the inside of the cell more negative, increasing the permeability to your sodium. So your sodium ions will open, leading to sodium influx. With that, this will trigger or this will start your depolarization. More sodium channels will open, more sodium ions will flow inside. Due to the inflow of your sodium, this will make the inside of the cell more excitable, producing an action potential. Then it will cease at positive 30 millivolts. The sodium channels would close or will be inactivated. By then, it will increase the membrane's permeability to potassium ions. So with additional potassium ions opening, this will lead to your potassium ions flowing outside, leading to your repolarization. If more potassium ions will leave the cell, making the inside of the cell environment severely negative, this will trigger your hyperpolarization. So it will be restored by your sodium-potassium pump, in which it will pump 2 potassium ions inside and 3 sodium ions outside. So this will restore to the resting membrane potential of negative 70 millivolts. Then lastly, you have your refractory period. You have your absolute and your relative. Your absolute refractory period is the short time while the axonema is still depolarized. A second stimulus, however strong, is unable to excite the nerve. This is when the sodium channels become inactivated and no stimulation will open additional sodium gates. Then you have your relative refractory period. It is the short interval during which the excitability of the nerve gradually returns to normal, or during your repolarization phase. So this limits the frequency and continuous excitation of the impulses because it will trigger the relaxation of your nerve.